Hi, Kelly. Professor Foster here. I just got your email and wanted to record a quick video in hopes of helping you understand operons a little bit better. So in your email, you said that everything you see says that using the right chemical will open the repressor protein, but it only seems to talk about lactose being the chemical that opens it. Would it be open for the proper glucose as well? Are lac operons a different thing than an operon that would use glucose as an energy source, or is it the same operon just switching energy sources? Any help is appreciated. Thanks so much. So first, let me thank you for sending your question, and let me try to answer. So really quickly, let's review the parts of an operon. Remember that an operon has three main parts. The first part is a regulatory gene. This gene sits just outside of kind of the business end of the operon, and it encodes a special protein called a repressor. So in this figure, the repressor is represented by this orange M. Repressors have a specific shape and amino acid sequence that allows them to bind the A's, T's, C's, and G's of this part of the operon. So this is called the operator site. Okay, the operator site. So the shape and the sequence of the repressor recognizes the DNA nucleotides in the operator site. The operator site is part of the second part of our operon. This is called the control region. You have the operator site, but you also have this site. Okay, so this is like a stretch of DNA nucleotides that make up something called the promoter. The promoter is the location where RNA polymerase binds the repressor. It binds the operon. Okay, and as long as there's no repressor here in the operator site, that RNA polymerase can transcribe these genes. Okay, these are the structural genes and they make up the third part of the operon. These are the genes that encode proteins that have a specific function. In the examples that you've been looking at and that we've talked about, one function that these genes might have is lactose metabolism. Remember, lactose is basically milk sugar. Some bacteria can absorb that sugar. They'll use these enzymes here to break that down to make galactose and glucose, which can be further broken down to make ATP, or energy for the cell. Okay, so with what you wrote, you said everything you see says that using the right chemical will open the repressor protein, but that it only seems to talk about lactose being the chemical that would open it. Would it be open for the proper glucose as well? So what you're saying is right. For lactose, you need for the lactose operon rather, lactose metabolism operon, you need lactose to come into the cell. That lactose has to find the repressor, it has to bind to the repressor, and then that lactose changes the shape of the repressor so it can't bind the operator site, and we get transcription of these genes and the cell breaks down lactose. Glucose gets broken down a different way though. Okay, so there's a process in the cell called glycolysis, and glycolysis is about 10 steps long. There's enzymes at every step that change molecules from one structure to another, and along the way we make energy for the cell in the form of ATP. Now, there's a difference between glucose metabolism and lactose metabolism. And the difference is the genes for glucose metabolism are produced constitutively. This means that the genes, I'm not spelling this right and I'm not going to waste your time. It's in my lecture notes, constitutively. Basically, what it means is that the genes for the enzymes in this pathway are produced at a constant rate all the time. So there's no repressor to turn those genes on and off. Okay, so it's different than what we see with lactose. So glucose cannot turn on operons or turn them off by binding repressor proteins. That's the first thing. Okay. Now, because the genes for glucose metabolism are constitutively expressed, that means that there are proteins for breaking down glucose always present in the cell. Okay, so any glucose diffuses in through a protein channel, these enzymes are able to bind it and start breaking it down to make ATP. But there are instances where the cell runs out of glucose. There's no more glucose around. The cell has absorbed all that it can from the environment. Okay, that's when those other pathways, those other operons for breaking down sugars becomes important. So when the cell runs out of glucose, it starts to starve and it produces a molecule called cyclic AMP. You can think of this as being like a panic hormone. The cell knows that it needs energy to live, but without glucose, it starts thinking, oh, I can't use that sugar for energy now. I've got to find something else. So cyclic AMP has the ability to bind another protein in the cell called the cap protein. And when these two come together, they are able to turn on some of these other operons, like the lac operon or the xylitol operon or the arabinose, what have you. So what they do is they bind the operator site. Remember, that's right there where the promoter is. Okay. And then once the cap protein binds the DNA at the operator site, it bends the DNA so that RNA polymerase can then bind. And we can potentially get transcription. But I know what you're thinking. You're thinking, wait, Professor Foster, isn't there supposed to be a repressor protein in the mix here? And the answer is yes. Ordinarily, there would be a repressor because we're talking about like lactose, mannitol, xylitol, and whatnot. Those are all sugars that the cell can use for energy, but it doesn't have to if glucose is around, right? So if there's glucose around, then all of those other operons are for the most part turned off. And usually that's because the repressor is binding in the operator site. Now, kind of like what you said, when specific types of sugar like the lactose or the xylitol or the arabinose or what have you, when that diffuses into the cell, it can bind to the repressor and change the repressor shape so that the repressor no longer binds the operator. That means that the roadblock that would normally be here is released. So RNA polymerase can then move forward and transcribe these genes. Okay, that produces a messenger RNA. The messenger RNA gets translated by ribosomes in the cell to make proteins that are capable of breaking down whatever sugar there is. So in a cell, you can have an operon for arabinose, and that operon will have its own arabinose operon repressor. Okay, so it'll have a repressor that turns off the arabinose operon. If arabinose diffuses into the cell, then it can bind specifically that arabinose repressor, okay, which turns on the arabinose operon so that the cell can now break that sugar down. What if it's not arabinose? What if there's lactose in the environment? Same thing. Different operon and a different repressor that's specific to the lac operon. So when lactose comes in, it's able to bind a specific receptor that turns off expression of the lac operon. When the lactose binds the repressor, then the repressor no longer binds the operator, and the genes for breaking down lactose get transcribed and translated. So the cell can suddenly start breaking that sugar down. And this is kind of the general theme. The arabinose operon has its own repressor. And when arabinose comes in, it binds the repressor. Repressor no longer binds the operator. Gene transcription can happen. Lactose has its own operon, and that operon has its own repressor. When lactose comes into the cell, it binds the repressor. The repressor changes its shape, no longer binds the operator. Transcription can happen from that specific operon. And I think it's really, really cool that cells have evolved this way, because the sugar that the sugar that gets broken down by the genes of a specific operon, right? It's the same thing that turns on the proteins for breaking down that sugar. That sounded kind of funny. Let me see if I can say it differently. Um, if the repressor is like a lock, okay, and the structural genes are like a house, you've got to unlock the door to a house if you want to come in, right? In this case, in our example, we want to metabolize sugar. The sugar is the key to the lock that opens the house so that metabolism of a specific sugar happens. I hope that makes sense. So going back to your question, so if I'm understanding your questions here, you're asking whether glucose binds a repressor on an operon as well. And I just want you to remember that the genes for glycolysis is how cells break down glucose, okay? The genes for glycolysis are expressed constitutively. They're always in the cell at a constant level. They're always expressed at a constant rate. Okay, so glucose doesn't necessarily open the operon. It doesn't bind the repressor to turn on an operon or to turn it off. Okay, the genes for glycolysis are always being expressed. Then you asked, are lac operons a different thing than an operon that would use glucose as an energy source? Or is it the same operon just switching energy sources? So lactose, the lac operon, is completely different than what we see for glucose. They're two different things. And if cells are able to metabolize other sugars, then they'll have their own operons as well. We don't really see sharing of operons per se. But that said, the metabolites from glucose, okay, they can turn on, they can be part of the system that turns on these other operons. So that's where we talked about the cyclic AMP. So that alarm hormone, that panic hormone that binds the cap protein, and then the cap protein binds the operon for lactose in our example, and that bends the DNA so that RNA polymerase can bind and we can get transcription. Okay, so there is some linkage that way, all right, but it's not that there's one operon that switches depending upon what the sugar is. Each sugar has its own operon. So the last thing I wanted to say is that, remember, again, there are different operons for all the sugars, but they can be linked. Okay, they can be linked to each other. There's research that suggests that there's a hierarchy of sugars that bacteria use when they're trying to survive. So glucose is kind of at the top. It's what they prefer to use. It's the easiest to use. It takes the fewest resources. Then comes lactose under that. And then there's a number of sugars that happen under lactose. So once the cell runs out of lactose in its environment, then it might turn on other operons. Okay. Now you might be wondering, well, it sounds like there's regulation for things like lactose and arabinose and mannitol and all these other sugars. Is glucose ever regulated? Glucose metabolism, is it ever regulated? So yes, it is, but it's a different type of regulation. So this little figure basically summarizes glycolysis. There's a lot more steps than what we see here. This just highlights the important ones as far as regulation is concerned. So what happens with glucose and glucose metabolism is there are some enzymes that are involved in glucose metabolism that can be bound by the products of glucose metabolism. So for example, the whole purpose of glycolysis is to convert glucose into ATP. Okay, and we get these two pyruvic acids along the way. ATP can bind fructose 6-phosphate and turn off all of the chemical reactions that follow, effectively turning off ATP production for the cell. Okay, so this happens in a few different places. You might remember from studying the Krebs cycle, maybe the citric acid cycle, that there's a molecule called citrate that gets produced. Citrate can also bind glucose 6-phosphate or phosphofructokinase and turn off glycolysis. So you turn off ATP production. So glycolysis is regulated also. So I know that was a lot of information. I hope that helps.